Hello students, welcome to the first lecture of week 6. So in the previous lectures we have looked at the basic requirements of assembly, what we achieve after assembly, like context, the concept of context, scaffolds, reads, paired end reads, how they are useful, how hybrid assembly is useful for the overall assembly, right? So now we are getting into the details of the methods that we are going to use in the various assembly processes, including short read and long read sequencing data. So we should separate them, we should assemble them separately, like short read separate, long read separate, and then merge, okay, or do the both together. Okay, so there are various ways in which we can do, and there are some background algorithms that are employed by most of the assembly software. Like for short read, there is one set of algorithm, that is de Bruijn graph. Long read, there is overlap layout consensus OLC method, right? So we'll be discussing these, and in this lecture I will introduce you to some of these methods in detail, and some I will touch today and will be covered in the future lectures. So basically, in the concept of assembly, just to again revise and to have a quick recap of what we did earlier. So basically, we start with a chromosome from which we extract DNA, and that DNA is sequenced into reads. Okay, now it could be short read or long read. So earlier it was mostly the short read data that used to be assembled, and basically the contact length or scaffold length was not much, or it is highly disjointed also, and but still, like, that was the only way, because short reads production was the only way, and it is also highly accurate, right? Then long reads came, and but they are suffering from errors. So okay, and they should be used along with the short reads, right? But recently high quality or basically error free long reads have also become available. So basically these things evolve, right? But at present the publications or the software that are commonly used, right, for short read and long read, basically they try to create two things. Either they create a reference based assembly where they remap the reads to a reference and generate a assembly. So if you have genome of a nearby species, or you are doing a resequencing of the same species for some purpose, including looking for some disease marker or some variation, or you are doing a different ethnic group in a population and like that, okay, or you are doing it de novo, like from scratch, you are assembling the contigs and reads, and making contigs, and still like going further scaffolds and the chromosomal level assembly, right? So basically after you have the de novo assembled genome, the other steps will be like you will be correcting the genome for any possible errors using either long read or short read data. You will also use the RNA-seq data because there will be because this data is coming from the coding region, that is the transcripts, right? So this will help you to correctly mark the genes, correct the start end of genes, polish the exon intron boundaries, and also predict the alternate spliced isoforms while you're doing the maker pipeline, right? Then basically along with these inputs, scaffolds are made, gaps are closed with the long reads, right? Then again, since you have used the long read, you can again go for another round of assembly polishing by short read, right? Because there could be errors. Okay, so by this repeated assembly and error correction, you will lead to a final genome assembly, which is taken for the downstream analysis, okay, which we'll be doing in the later part of the lectures. Okay, so like the overall summary remains like you have long read and short read. Long read basically by Peck-Boyer ONT, Oxford Nanotechnology, short read by Illumina, right? And there will be algorithms or software or tools for assembly which are dealing with either the short read or the long read, and there are some tools that deal with the hybrid assembly, but for that basically you need to do the assembly separately, preferably, right? And then after you have a good assembly by hybrid assembly by these methods, you can go for high C or BioNano to go for the chromosomal level assembly. Okay. So now which tool should be used at what stage? So basically for the short read, either you are using Illumina or using 10x chromium instrument to generate link reads, right? And then sequencing by the Illumina technology only. So these are all short reads, but here it is mainly the link reads, and here it is the randomly fragmented shotgun data, right? So for this, the assembler for 10x chromium, it is Supernova, right? And for Illumina, there are various assemblers, Velvet, All Paths, came earlier, but IBIS is one of the commonly used, and which we'll also discuss today. All these are based on De Bruijn graph, Supernova, IBIS, right? For short and long read hybrid, basically Masuka or DBG2OLC can be used, right? This we will deal with the later classes when we do the hybrid assembly part. And for the long read, either like PacBio or Nanopore, Kanu, Falcon, Flye are the software, and we will be discussing Flye today. Okay, so the genome assembly algorithms are broadly divided into two types, or basically there are two types, right? The De Bruijn graph approach or the overlap layout consensus method. So this is for the short read data, and this is for the long read data. Okay, so the basic approach of a De Bruijn graph is that it will be dealing with the K-mer. So the choice of K-mer while you are assembling is very important, and it can also get this information from some prior work like Jellyfish or other software, or it can also iteratively run on different K-mers and produce you different assemblies, and then you can select that at which K-mer you are getting the better assembly, right? So basically K-mer is nothing but a subset of your read, okay, of a particular length. For example, your read length is 150 base pair, so K-mer length could be like ideally rough rule is that half of your read size plus some base pairs, like it could be like 81, 91, 101 or 111, like that. Okay, so then it arranges those K-mers into a hash table, right? And basically those K-mers are generated in a stepwise fashion, such that each K-mer is preceding the previous K-mer by one base pair, right? And this way the entire range of K-mers is generated from a read. Okay, and then basically based on their frequency and the next K-mer that matches that K-mer and exceeds that K-mer by one base pair, they are arranged together and the tree is traversed, so that the hash table is traversed, so that you can generate the assembly. And when you start seeing the K-mers which are assembling further, you will stop, and that will be a quantity. Okay, like this. So this is the read from which these K-mers are derived. So just for representation purpose, the K-mers are 3 mers like TTC, TCA, but there are these will be very long in the real scenario. Okay, like more than half of your read length. That means like if read length is 75, then it should be more than that. Okay, so the chances of randomly finding these K-mers will be really less in genomes, because see, for example, primer length you choose something like 16 to 25 base pair, thinking that it may not ambiguously match multiple regions in the genome or other genomes. Similarly, like K-mer, if you are choosing like more than 75 base pairs, so that will be a very good length for randomly. Even like more than 25 base pair, anything will have very less chances of finding randomly in the genome. Okay, so basically from any read, K-mers are derived like this. So TTC, TCT, CTT, CTA, TAA, AAG. So okay, so in a overlapping fashion. So from all reads, these K-mers are derived and they are arranged in a tree. Okay, so basically the K-mers becomes vertices in the De Bruijn graph. Okay, like this. Okay, and which are connected with edges by the overlapping K-mers. For example, now when we are arranging these K-mers, we will start building the graph. So we will see the frequency of CGA, then exceeded by one more GAT, then ATT, then TTC, and like that we will keep on building. And at places you may find that it may branch out because there was heterozygosity or some error, or possibility that it has two branches. So those may confuse the algorithm, and many times like you form unitics which stop at this level, right? So that you don't go into the branching process, and that is the end of your unitic. Okay, so basically this graph is walked through and you get the consensus sequence, assembled sequence, right? So recently like several tools are available like Spades, Velvet, SOAPdenovo, Megahit, Abyss. Okay, so all these are very promising, and Spades we'll be discussing further, and Abyss is we are discussing today because this is one of the commonly used tool, right? And so basically this is based on de Bruijn graph, and the concept I have told, but there will be some changes in the de Bruijn graph implementation also, that we will see in this Abyss assembler. So recently the new version is Abyss 2.0, which is much more better than the previous Abyss assembler, because the groups who have produced the previous one have seen the problems and issues and scope where they could improve it, and they have improved it using a Bloom filter based approach, which we will discuss today. So this can be a very good approach to assemble the large genomes from de novo assembly using short reads. Okay, so basically it's a multi-stage de novo assembly pipeline, you can say, which can form unitics, contigs, and scaffolds. So you can choose like which you want to go for, right? And basically at the unitic stage, it performs the initial assembly of sequences according to de Bruijn graph algorithm that we will see today. Okay, so basically it takes the set of K-mers, derive the overlapping K-mers from the reads basically, and store that in a hash table. So we will see in the hash table. So there will be some K-mers which it will found from the repeat, and some it will not. Okay, so basically it will not waste time in the K-mers which is not found. So it will look at the presence and absence of possible neighbor K-mers in the de Bruijn graph. Okay, and then only those which are present will be assigned 1 and other will be assigned 0. Okay, and then it will go for basically constructing the hash table, which is Bloom filter, and then traversing through that hash table. Okay, so this is the way it does. So from this read basically it derived the various K-mers. So again, like these are 3-mers, but in real situation they will be much longer. They will arrange them in this hash table. Okay, so basically you can see that based on their occurrence they are arranged. So those who are found are labeled 1, like TGG is found, so it is called 1. GCA is found, is called 1. Okay, and those which are not found, okay, or not possible from these set of reads or K-mers in the overlapping fashion, so they will be assigned 0. Okay, and then it traverses through that Bloom filter. So exceeding by 1 base pair, finding the K-mer which best matches the previous K-mer. Okay, but exceeding by 1 base pair, right? This way it will keep on moving through, right? So and basically these were not present possibilities like CAA, CAC, all 4, like basically after CA, GCA, what can come next? CAT, CAG, CAC, CAA. Okay, so it found CAG, okay, which was there in the hash table, and it will proceed with it and connect it. Okay, so basically by this approach the overall memory requirements are much lesser, which is one of the limiting factor when we are doing the large genome assembly. Okay, because we are dealing with maybe 200-300x data for a genome. So consider human genome, you generate like 300x, 200x, so that is 600 GB data you are taking. So that may take like more than 600 GB of your RAM, which is not available with almost everybody, so only with large groups. So that is why these Abyss can still run on smaller RAM also. Okay, so once it has made the solid state, it will extend it in both directions. Okay, and whenever it sees a branching, for example, it could have found both CAG and CAT here, so a branching kind of thing, so it will stop at this level, so it will truncate it at this level, right? and will create the unitig at this level, right. So this is the concept of ABIS. Okay. So basically this again to K-mer from each input sequencing read are loaded into a Bloom filter as we see in the previous one. Okay, and basically hash values of each K-mer are computed. Okay, and then basically you will this de Bruijn graph is traversed by repeatedly querying for possible successful successor K-mers. Okay, so like this was one K-mer and the next K-mer will be like exactly matching the previous one, but one more in an overlapping fashion, like one this way, right. So this is the next K-mer which matches exactly the previous one but exceeds by one base pair, right, and this way it will be single base pair extension, it will extend in both directions. Okay, and this is then extended in both directions, and it breaks whenever it finds a branching pattern or place where there are more than one possibility of finding the K-mer in two directions. Okay, so the for example, like it is written, extension of solid is halted when either a branching point or a dead end in basically it did not find another K-mer which is overlapping to the previous one, or it is find two K-mers which can generate the branching point, so it halts over there. So for the installation, basically GitHub is again the place which is the source of almost all the software, right, and biology related software. So basically you can download it from GitHub, right, and then there is always a readme file. You should read that file which gives you the instructions to install, but just for your convenience, basically there you will run a script called /autogen.sh, and generally these scripts are executables. Otherwise you can change permission chmod +x and say autogen.sh, and then you can run it. Then you say make directory build mkdir build. Then once it is the directory is built, you can go to cd build and then do the /configure. That means you are configuring this for your system. Okay, -1 prefix, and the path of ABIS where you are configuring it. Then basically they say make and make install, which is the common way of installing the programs. Okay, so most of the program follow this way of installation that we have also seen in the when we are doing the Linux and how to install the biology related software. Okay, now the command line for this is you can always get help for ABIS -PE, and then you can use any of these additional parameters, but the parameters that are standard are basically ABIS -PE. P is for the paired end reads, because you'll always be using the paired end reads. Okay, most of the cases those are more helpful also for assembly. Then any name of assembly you want to define, it will call your assembly by that name, right. Then the K-mer size. Okay, so it could be predetermined, or you can start with 1 and exceed by 10 mer. Okay, like 91 or 96, 86. Okay, you can go in both directions and see, like if it is increase in 10 base pair, like 10 K-mer length like 96, then go for 86 and then go for 106. So by going in this direction or this direction, choosing a bigger or smaller K-mer, if it is increasing your N50 and decreasing the number of contigs and covering your genome well, then you will choose that length. Okay, you can do that iteratively by a script also. Okay, then b equal to 2g, that means what it should be the Bloom filter size, that means the hash size. Okay, so basically it depends upon your RAM. Okay, and you can use any of these. So basically here it is 2g, that means 2 GB. Okay, and input equal to read 1 and read 2. Okay, that means forward and reverse read, the paired and read. Forward read table file containing multi-FASTA sequencing of the forward reads and corresponding reverse reads multi-FASTA. Okay, so basically the two files should also have like all the sequences, and number of counts in the two files should be same, right. So the choice of K-mer matters a lot, and Sometimes by experience you can pick a K-mer and then go both sides and just to check whether it is performing the best, or you can do iteratively, right, by running this in a script, this command, right. So basically it is recommended to use odd values of at least half of the length of read. So as I mentioned, basically if the length is 150 base pair, so you can go for 75 plus minus 10, and you can go like either way and see whether decreasing the K-mer size or increasing is affecting your assembly size. Okay, then it will, you can specify like you want to generate the unitic file, okay, which is whenever it sees a branching it will stop, or the contig file, right, or scaffolds file or long scaffold file. Basically all will be generated in FASTA format, okay, or you want to save the graph files, or okay, and what folder you want to save it. So these options you can provide. So the output will look like this. So it will give you the assembly statistics at various length of contigs, like more than 0 base pair, like all the contigs. Like basically if you see the file, at the bottom of the file you will see several contigs, or which are much smaller than your read size also, okay, which could be like K-mers, okay. So you should get rid of them. So basically 1 KB and above contig, if you see like unitics were 10,840 and contigs were 970, because contigs are made from the unitics, right, and the assembled scaffold was about 7,000. But when you go for more than 5 KB, so basically you could get unitics you did not get because it was too long a size for the unitics for short reads, and assembled contigs were 93 and total scaffold were this. But at this length, if you see, basically the length of genome that you are covering, okay, is also decreasing. Like if you connect this, right. So basically this is only 4 MB, okay, whereas at 1 KB it was 22 MB. So if you increase the length of contig, okay, so you may be compromising with the total coverage of your assembly. So you should know, for example, if you know that your genome is like 5 MB and you are able to cover like 5 MB by 5 KB plus contigs, then you can select that, but you may be losing a lot of information in the smaller contigs, right. And like for contig greater than 500 base pair, it gave 60,000 contigs, unitics 53,000 unitic, okay. And at this length, basically it also tells you like GC and 50. So 500 base pair and above contigs is a good length, because the read wise size was 150 base pair. So 500 base pair is about 3 times more than your read size. That means some assembly has happened in the reads that you have taken, and there were 2 reads, forward and reverse. So at least you expect, like if they overlap, if the insert size allow that, okay, then you may achieve like slightly bigger read size, or basically assembly of the forward reverse read, okay. So basically you can see that 500 base pair is one of the preferred minimal contig size, and at that contig size, basically the N50 was only 789 base pair, okay, whereas the total length that was covered was almost 52 MB. That means it the total length of all the sum of all the contigs was 52 MB. That means it might be able to cover your assembly better, but it will be highly fragmented, okay, in about 51,000 or 60,000 pieces. Okay, so basically ABIS2 has an advantage on others. Like if you see, like RAM wise, ABIS1 was taking 418 GB of RAM, whereas ABIS2 is taking only 34 GB for that purpose. Time may be little longer, but basically at the memory wise it is much smaller, and compared to other software which like SOAPdenovo, okay, this one does not perform very good. So we should ignore this for now, okay. It is much lesser than the other ones. Then basically SOAPdenovo is another short read assembler based on deep Bruijn graph approach. Mostly it is the basic methodology is similar, but it has some differences. Okay, so it is specifically designed for Illumina short reads. Okay, and it also uses the de Bruijn graph approach. Okay, which does the contig assembly, error correction, paired end assembly, scaffold construction and gap closure. So which is the advantage of SOAPdenovo. So in the version 2, basically it has improved the error correction module. It has sparse de Bruijn graph is available in these SOAPdenovo 2. Improve contig building, improve scaffolding, improve gap closure. So basically the newer version of any software will be an advantage or improved version on the previous one. So you may prefer to use the newer one, right? And basically installation wise also that may be a better fit to your new OS, right? So if you see here, like N50 or N50 is, as we explained earlier, is the point at which basically half of the length of contig, above which you can cover almost half of the genome, right? So N50 should be as higher as possible, and the number of contigs that are remaining after N50 to the end should be as low as possible. That means that in minimal number of pieces you have covered the assembly, and also the N50 or basically the contig sizes are much bigger for that assembly, right? So here the N50 for SOAPdenovo was 7.9 KB, whereas that improved almost 10 times in case of SOAPdenovo 2. Okay, number of contigs decreased 10 times. Okay, and which is a huge and basically N50 of scaffold was 1.3 KB, which is a huge advantage on the previous SOAPdenovo version. Okay, and this is on a sample genome boss impatience, right? So to install it, you can get it from GitHub. It is a tar.gz file. So you'll unpack it by tar -zxvf and this file name, and this will unpack it in your directory. Okay, and then basically based on the instructions, you will install it again the same way you have installed the previous ABS, right? And this is the configuration file where you can specify like the K-mer size, how many threads you want to run your system. Okay, the what to do with the low frequency K-mers and very high frequency K-mers, right? And okay, so basically I am giving you an example of configuration file in the next page, but basically the command runs like this: /SOAPdenovo all means it has 4 steps, right? So either you can run all the 4 steps separately by specifying that step name here, or you can say a wrapper script which is say all, which will run all the 4 steps. So basically we go for all, but I am also showing you slides where each step can be used, what command can be done for that. But so that may be helpful sometimes to debug or basically know that what is the outcome at each step. But I think I would go with all and use the script that is running all the subscripts in one go. Okay, then -s config file. So this is a config file that we will see in the next slide what it contains, where you define all the parameters and everything. K-mer size, okay, and then the output file. Okay, so this is a config file. You can say the what should be maximum read length. So your read length may be 150 base pair, but you might have trimmed some bases from either end or remove the low quality bases. So you may end up having a variable read length. Okay, so you may choose some of the minimal read length that you want to take. Okay, average insert size, what was like when you are doing the library preparation, that you have to specify, right? And the other parameters that are required, okay, for to running this program. So basically you may keep the other options things are default, because at default generally it is optimized to run best at the default parameters, right? So okay, so this is all and config file and the K-mer size and the output. and if you want to run step by step, then there will be 4-step pre-graph. Okay, that will make a pre-graph of the K-mers. Okay, make it, assemble the contigs, map them, and prepare the scaffolds, right? So this is the main output that you will get, a contig file and a scaffold file. So in the FASTA format, right, which will you can use for further next step of assembly is polishing or hybrid assembly, right? Okay, or you from the different steps, if you run like pre-graph, so you'll get these files, right? K-mer frequency, edge, etc., etc. From the next step, when you say contig, you will get these files. Okay, I'm not going into details of this, but just showing you that you may get a lot of files, and it may be confusing for you if you are doing this for the first time. So even we do not do this, so don't go for the command to run for each of the steps separately, and map, and the last one is scaffold. So basically a bunch of files will come, okay, which you will be taking to the next step like this. So either you can choose this command or you can do step by step. So preferring this command is a better idea. Okay, then the next one is the long read assembly method, and for that the overlap, layout, and building the consensus or consensus OLC is the name for the algorithm. Okay, which is not dealing with like K-mer like of approach. Okay, but it will be mostly like the reads in the long, because they are already long reads, so it will try to find the overlap between the reads and organize the read according to the overlap, right? And then try to build the consensus based on the overlap, because there could be more than one long read which may be mapping to that layout of reads. So looking at what is the consensus base pair at every position, and based on the occurrences or frequency of reads in that region, okay, it will build the consensus. So it is the preferred method for the long read assembly, and because anyway this long read assembly is going to be corrected by using short reads later. Okay, so one of the common and most used software is the Flye assembler, which gives very good output, and it is the method of choice here for the long read assembly, right? And it was published in Nature Biotechnology in 2019. Okay, so it's a sequencer for assembler for the PacBio or Oxford Nanopore long reads, right? And before using this, you have to clean the reads by the things, the previous classes we have discussed, like how to clean up your reads to prepare them for assembly. Okay, so it can take the PacBio ONT reads and provide you the assembled contigs as the output. Okay, it uses a repeat graphs as a core data structure. Okay, and basically it starts with a single read, try to find the overlap. Okay, and then basically prepare a layout of all the reads and then build the consensus. Okay, so it has different modules to do each and every step, right? Then next we'll be doing the Flye software in detail in the next lecture, so I am not going into detail of this. I will introduce another long read assembler, that is Supernova. It's basically not a long read, it's a short read assembler but link read assembler. Okay, so link reads have been used in recent times, which are generated by the 10x chromium library preparation. So 10x is an instrument. Okay, and basically the idea is that the long distance information okay, or the reads which are linked by and separated by long distance. So we have seen like how the library is prepared for this in the previous classes. So basically that data now we are analyzing to build the assembly. So either you can use the 10x data if it is at least 60 to 70x of your genome, right? And basically these are short Illumina reads only, and that may be sufficient to give you a good assembly, right? Otherwise you will be using the Illumina data with like little higher assembly, a little deeper coverage or more coverage and more X, okay, and but here like using link reads, like lower coverage should also be okay for that, okay. So basically good thing about Supernova is that it can also create deployed assemblies and separating the maternal and paternal chromosomes, okay, also, right. And so basically it will be taking the 10x data, the short read Illumina data with link read information, right, and it will be using that for the generating the contigs. Okay, so this Supernova also we are going to cover in the later classes, so we will not go into detail of this, but Supernova is the method of choice and basically it is provided by the 10x genomics company only. So it is their software that they provide for the genomic assembly of 10x reads, and it contains a set of Python scripts called proc10xg, okay, which is which has basically two components, that is the process and the filter component, okay, basically process and filter scripts, right, which do this assembly in 2 steps, okay. So it can take data either in BCL to FASTQ format or FASTQ format, Supernova FASTQ format, right, and then basically it assembles and give you the contigs. So system requirements are like minimal for this, and also depend upon how much data you are dealing with, but a simple like 16 core AMD or Intel processor, 500 GB disk space and 256 GB RAM is required for this kind of assembly. And we will be covering both Flye and Supernova and SOAPdenovo also in the later classes. So I am not going into details of this in this. So basically, okay, so basically this was also published, the details about the 10x technology and the software in this paper. So all these assemblers should take care of the basic problems that we deal in the genomes, that is the heterozygosity. More heterozygous the genome is, more problems there would be in assembly, and you may require more data or like more amount of longer reads to resolve the heterozygosity. Similarly, ploidy level, which is a typical problem in plants genomes, and again like long read and short read, a combination of that is preferable, and the software that you are using should be able to resolve it. And similarly for repeats, you will definitely need the long reads and the long read assembly algorithms to sufficiently cover or correctly cover the read region along with the flanking regions, so that you can put the read in the genomic context at the right place and build the scaffolds. Okay, so this is good for this class, and this class we covered basically we introduced the commonly used short read and long read assemblers, and went into details of ABIS and SOAPdenovo in this, and Flye and Supernova are introduced here but will be covered in the later series of lectures. So so basically you get an idea of how these assemblers can use the short read data, what algorithm they use to assemble, and basically newer versions of these are coming, which you can use to get a better assembly quality. Okay, so thank you for today.